plan for today is to go over concepts of applied and industrial. Yes. And then on Wednesday, we're going to review for the final. A good chunk of Wednesday's lecture will be review. And so the actual final exam is Wednesday next week from 2 to 4. Okay, next Wednesday. Viruses, right? I think that's where we start. Viruses onward. We already did the fungi, didn't we? Okay, so viruses forward. Wednesday or Friday, when I just meet with you in lecture, I'll be able to tell you more about the final exam. I did mention there might be a couple of topics from cumulative concepts. If that's the case, I will give you the topics. And because I don't know yet, I have to look at the data. So I have to look at the stats again and decide if there's going to be, there'll probably be at least 2 questions that are cumulative, but I'll let you know the topic. Generally, when I do cumulative stuff on the final exam, I tend to make those more like multiple choice or matching. And I say, like for the essay portion of the final exam, usually the essay questions would be new stuff that you haven't been tested on yet. So just keep that in mind. So the last topic is industrial and applied. And boy, this is a huge topic. So I am definitely scratching the surface. I'm hoping by the end of this, you'll have a better appreciation for how we use microbes in so many different ways to improve human life and other animals. So hopefully you'll get a better appreciation for that. But the applied and industrial, what we're looking at is, the different ways we can work with microbes or their products in order to benefit some aspect of our health or society. And so here you're just seeing a big picture idea of ways that they work with microbes and things that they might be doing. Of course, yeast cells, we all probably immediately go to, oh, well, they're used for fermentation and brewing beer and wine and all of that, but they do a lot more than just that. And then I'll be working through some specific examples of how microbes are helping us. So this is just a partial list of, usually when you think about microbes and industrial uses, a lot of people can easily pick up the idea of alcohol production, yeast involved in that, but we can actually eat yeast too. Just eat them. Certain strains of yeast are very healthy, and yeast are used for a lot of different things in alcohol production. I'll talk a little bit more in detail on antibiotics, vitamins as well, and amino acids. But like I said, I'm scratching the surface. There are so many uses of microbes, and it just keeps going and going and going, especially now with the field of synthetic biology. Have you heard that phrase, synthetic biology? Sometimes they shorten it. They say synbio. Synbio. And that is a really hot field of study right now. Every year they have this big conference out in California. This one society that puts it on, and it brings entrepreneurs and people from all over the world that are using synthetic biology to for a certain particular goal. I wanted to go to it because a lot of it I could use in my teaching. And entrance fee to get into that. Conferences are already expensive. Just if I go to a regular microbiology conference, it can be $700 just to register for the conference. This one was over $2,000, I think, something ridiculous just to register. That doesn't include airfare, hotel, food. But okay, I'm not going to that one, but unfortunately. So anyways, so if you want to work with microbes, There are certain things that you need to consider that either for safety reasons or for profitability, you want to make sure that they have certain characteristics. High yielding, that's good for profit. Whatever thing you're trying to get the microbe to do, you want them to do it a lot. And you want them to do it quickly if possible, which is usually not terribly hard to do because microbes tend to grow very quickly. And if you get to know your microbe and optimize their growth conditions, you can really accomplish high yields in short times. Often what you do is you start small and then you discover something and then you want to scale up and see if you can make a profit off this, which means you have to go big. Has to be able to deal with that. And we'll talk a little bit more about that. Often the preferred way to work with them is liquid cultures. It's very much, well, it's more straightforward. It's easier to work with. It's usually cheaper. They often use the phrase scale up capabilities. That just means you want to be able to take the microbe that you discovered in a test tube or a flask and be able to take it all the way to huge fermentation tanks, perhaps, or some kind of huge scale up system. Spore producers can be very nice to work with. That was one reason why I was very drawn to working with Streptomyces bacteria because they can produce spores. And so I saw that if we could find good biocontrol agents, we should be able to provide that into the commercial realm because if you produce spores, they have stable shelf life, right? And so it would be a way to bring this idea to market and actually get it out there in the real world if they're capable of producing spores. They're also easy to work with. If they form spores, you can just harvest the spores and freeze them. So you always have a backup microbe to work with. They're just really nice. They're spore producers. But, you know, there's only so many microbes out there that are spore producers. So not everyone can do it. Growing rapidly is good. Not a pathogen is a definite plus, especially if you're the worker working with them day in and day out. And this is a nice idea too, especially with synthetic biology, capable of genetic manipulation, where you might want to tinker a little bit with what they are capable of and maybe enhance it or delete out a gene that you don't really want them turning that one on. So let's just get rid of it. And so this idea of being able to manipulate them, and again, in the realm of synthetic biology, that's a big aspect that they consider. This is taken from a published resource on industrial biotechnology, and it's saying a lot of what I just said in the previous slide, but there's a few extra things in here too. So in general, when you're studying this, being able to either recognize a good characteristic or be able to talk about it if I ask you an essay question, be able to give some examples of what makes a good microbe for applied or industrial uses. So that very first one, it says it must be contamination free. That's kind of a no-brainer. And then they say it should be phage free. I mean, so again, getting at this idea that if there is a bacteriophage in there, they might have brought along some genes that in some cases it can make ordinary bacteria become toxin producers. You know, we did talk about that. So being phage free is usually a good thing to think about. Okay. All right. This is a classic example of looking at production and whether or not you're aiming for a primary metabolite or a secondary metabolite. So for example, on the left, you see the green line is showing alcohol production. So when they ferment yeast and want them to produce alcohol, that's actually a primary metabolic pathway because that's the whole idea where you give them sugar, they go through glycolysis, they take it to pyruvate, and then you deny them oxygen. So then they take it down into the fermentation products and you shift it so that they're making alcohol. So it's very classic that you see this pattern where you will have the cells increasing. So this is like a growth curve of the yeast in the red line. And then you can see that they're consuming the sugar as they're growing. So as their populations increase, they're using up more of the sugar. So the sugar goes down. And then you can see the alcohol is pretty much in tandem with the cell growth idea. That's classic for primary metabolite. Now on the right, they're showing the idea of secondary metabolites. So secondary metabolites are non-essential for growth. And you kind of have to know your microbe to figure out a way to get it to do this. And so it's a little bit more detective work or really getting to know your microbe up front if you want them to produce a secondary metabolite. They don't do it all the time, like with the primary metabolites. They don't do it all the time. And you have to give them the right nutrition to kind of trigger it and the right situation to make it happen. Often, if it's a spore-producing bacteria, they'll often shift into some secondary metabolite production as they form spores. Like the Streptomyces bacteria that I work with, that's what they do. As they shift into, okay, I'm going to make spores, they ramp up their antibiotic production. And they think that the microbe is doing that because you shift into spores, that phase of your life, into spore production, you're kind of vulnerable as you're doing that. And so pumping out some antibiotics might be a good strategy. That's one hypothesis. So the right graph is showing kind of a classic idea of secondary metabolites where you have the microbe growing and you can see their growth curve on the red line. They're eating up all the sugars, so you see that going down over time. And then you see a lag in when penicillin is being created. You don't see it kind of going in lockstep with the cell growth curve, with the cells there. And so what you see is a delay. And it's really not until they get kind of, late stationary phase. If you look, that's about late stationary phase where I just circled it. And that's when you start to see penicillin increasing. And that's very, very common. When I grow my Streptomyces bacteria and I'm hunting for antibiotics, I go home and take a nap when I see they're in early stationary. And then I have to come back and sample, and sometimes through the night sample and get the plates. And I see them really pumping out high doses of antibiotics. So this kind of idea, you have to understand what you're hunting for, what you're going for. Is it a primary metabolite or secondary metabolite? And I would argue that secondary metabolites are a little trickier because they're only produced under certain conditions. And you got to know what those conditions are. So often you have to grow them in controlled systems where you really limit the risk of contamination from just things flying around in the lab that you're working in. And you can get pretty simple systems or a little bit more sophisticated like this, but you can see that both of these are kind of showing the same thing. You control the heat. So you usually have like a jacket on there with water running in there to cool it down and get it at a precise temperature. You can control the pH pretty easily. You have inputs and outputs, all sterile, aseptic. And then you can see very often you have to stir. And it depends on the microbe. That stirring might just be to get the broth evenly dispersed and all the nutrients in it just evenly dispersed, or you might be stirring it to aerate it. It just depends on the microbe. With yeast, if you're trying to get them to do alcohol production, you don't want a lot of oxygen in there. So you don't try to aerate. You just want it stirred up a little bit to make sure nutrients are getting to yeast and they continue to do their thing. But like with the Streptomyces bacteria I work with, they need a lot of oxygen. That's one of the downsides. They're kind of a highly aerobic microbe. And so you really have to stir very vigorously and you need a lot of headspace, we call it. There goes some headspace so that there's oxygen being drawn in, and then with the rapid stirring, it's getting mixed into the broth. And then just remember, oxygen in liquid dissolves pretty slowly. So that's another reason why you really have to speed up the mixing. So when I have my platform shakers in my lab, when I grow my Streptomyces, they're zinging around on that platform shaker at like 250 revolutions. And they're just, it's very obvious. They're being vigorously stirred. Whereas most of the bacteria we grow for you, instead of 250, we drop it down to like 170, and that's totally adequate. Sterile, keeping the right heat levels. Often all of this heats up things, so you have to use like a cooling water jacket to cool it down. And then aeration may or may not be necessary, depends on what you're going for. So you can have both anaerobic or aerobic. So they call them fermenters, which usually you go to, well, that should be anaerobic, right? Yes, but they also call these types of apparatus, they generally say the fermenters. Sorry. So it's a little weird, but so they can be anaerobic or aerobic. But basically, the scale up idea, this is showing very much a large scale operation, but often discoveries are made in test tubes, flasks, and then you start like a benchtop fermentation system, and then you just keep ramping up and scaling up inside complexity. But they have the technology now where it's not terribly difficult. You do need some physics and chemistry to learn how to work these, or have a good team that can take on those roles of a good interdisciplinary team to make sure everything's going okay. But when you look at some of the scale ups that they've done, this table's from your book. It's nice. I'm not going to ask any details on this, but I think it's good for you to look at the basic idea, because I really like that they put in here, like, how did they used to do it? And now with the help of microbes, how are we doing it today? And then what has that done? How have we benefited from switching into using microbes? And over, you could see that when they started using microbes for these different processes, it really did help quite a bit, many different ways. So in general, if you could be able to like talk about an example of this, like what it would be a good example. This table has enough information where you can pick one and talk about it. I like the one down low, the detergent one, because this is a big deal. Phosphates for cleaning and brightening, and now they use bacterial and fungal enzymes for that purpose instead. So they've been able to replace phosphates with microbial enzymes, and that has gone a long way to decreasing water pollution. And our water is so precious. We've done so much damage to it. So this is a huge advance, being able to use microbial enzymes. But you can see there's other ideas here too. Even with diapers. I didn't know that. Bacillus ferments the corn sugar to lactic acid, which is heated to generate a biodegradable polymer for woven fabrics. Had no idea. Biodegradable diapers, that's awesome. And then stonewashed jeans. Ever wonder how your jeans got stonewashed? It's microbial enzymes again. That's the new method. The old method was quite toxic and nasty. And now they just put in enzymes as they're soaking the denim jeans to make them look stonewashed and well broken in. And then here is another table just showing, kind of grouping it by different classes of products. Again, you can see all kinds of uses, agricultural, the gibberellin, plant hormones. Whoops. insecticides. They use, yeah, live pathogens, biocontrol essentially. But yeah, the list goes on and on and on. And then they show the different bacteria and fungi. So fungi are really a group, fungal group as a whole. They're getting looked at more carefully for applied purposes. And I think later in the slide set, they also are getting much more into growing groups of microbes in order to get a particular product or thing to occur. And I'll talk a little bit more about that. So now instead of just growing everything in isolation and in purity, they're starting to look at synergy between microbes and finding some really cool things going on with that. So antibiotic discovery has been a long process for decades now. And if you look at this list of who's doing it, the microbes, you can see streptomyces are showing up over and over. So chances are, I would wager a hefty bet that most of us in this room have had multiple antibiotics made by streptomyces bacteria. And that's how they still do it. They don't synthetically make these. It's easier to grow the microbe and make them do it than it is often to try to synthetically make it. So with the streptomyces, I just put this in here to refresh your memory. When you look at them, they're different than the typical bacteria you've been working in the lab because they look more like a petite fungus with hyphae growth, and then some of their hyphae come up off the outer surface and soil and they start to fragment forward. So that you're down on the colony here in the top right picture, and all that black oozy stuff that's oozing out of the colony, those are pure antibiotic crystals that have oozed out. And so the idea here is if you learn their growth cycle, their life cycle, you can capture their antibiotics. You can test for them too. If you're looking for new antibiotics, it's very common that you'll do tite assays. And here, this image, figure A, what they did there, it looks like they're running what's called a HERR assay, HERR. And what you do is you take a soil sample, because most streptomyces are from the soil, and they are still doing this around the world. They'll take soil and they're hunting for other streptomyces bacteria. And you do a dilution and a spread plate, and then you mix your pathogen that you're trying to see if you can find a new antibiotic against it into like some soft agar and pour that over. And then you incubate. And so all these colonies showing up are from the soil sample. Where you see clearing, that's where the pathogen was killed off. And so this is a very, like one of the initial things you would do if you're hunting for antibiotics, you take that soil here assay, which is basically just a spread plate where you challenge the pathogen against the colonies that are going to start growing. And then after that, you can actually go in with a needle, take it down, pass the soft agar, grab to produce the organism, and start looking at it, purifying it. And so then some of these studies that you see on figure B, this is the streptomyces bacteria, the whitish colony that's put down kind of as a lawn on the agar plate. And then these streak lines are the bacteria you're trying to see, is there anything that strep is producing that can inhibit these different bacteria? And so on one plate, you can test multiple sensitivities from multiple bacteria or pathogens. And so this idea, once you find something that looks promising, then you try to extract it, purify it, and try to figure out ways to optimize the production of it. It's a lot of work. It's also, just because you find an antibiotic doesn't necessarily mean we can put it into use because it might be too toxic to the human or the animal. So you have to be, it has to go through a lot of trials and has to pass all the governmental laws in place for not creating any harmful situation. So it's a ton of work. It's a lot of money. to go into antibiotic discovery. You put in a lot of money to do this, and that's why a lot of companies right now are phasing out their antibiotic discovery aspects of their business. And so that has scared a lot of people, because if they're not going to do it, they're the ones that are pulling in the money from other drugs, right? The drugs that you take on a more regular basis, the profit margin is high. And but they say it takes so much money to get an antibiotic to market. And then the other thing, think about it. When you go and get an antibiotic from your doctor, how many days do you end up taking it? What's been a normal thing for you? How many days in a row do you take it? 6 days, 10 days, and then hopefully you're cured, right? So you stop taking it. So the profit for that company, if you only take it for 10 days, they're not going to make much on it. Whereas if they make a drug like, a heart medication, where you have to take it every day for the rest of your life, that's a much better option from a profit margin idea. So there's been rumbling going on that they may shift antibiotic discovery into the government system to see if, kind of take the profit margin out of it and have other entities take over this role, because the businesses are starting to phase it out. So just be aware of that. To me, that's super scary that we don't have enough people working on this. And that's one of the reasons why I'm so excited about quorum sensing. I think that might be another avenue. There's other avenues besides quorum sensing, but I think that's a nice alternative to use of antibiotics and antibiotic resistance. So just be aware of that. Vitamins. Did you know B12 is made by microbes? And you have to have B12 in your diet. And it's the only way that we get it is through microbes. We can't make it. Our cells cannot make B12. We have to get it. And the ones that they're using commercially are Pseudomonas and Propionibacterium species. But look at that, how complicated. Largest and most structurally complicated vitamin. Neither fungi, plants, nor animals are capable of making B12. Only bacteria. So very necessary vitamin that we have to have, and it's only done through bacteria. Yeah, I would not want to be the chemist trying to synthetically make that thing. Looks pretty complicated. Amino acids are a big business, making those and using them for a variety of things. So here you can see different reasons you might do add it into foods for flavor or antioxidants, nutrients. Amino acids. Have amino acids in their diet, certain nutrients, but they also improve certain foods. Although some on here I'll mention more detail, maybe aren't so good for us. But let's look at they have on here lysines on here. So let's look at lysine a little bit. Lysine is a good example of getting to know your microbe and understanding that you need to tweak it a little bit. So here is the production of lysine from spartate. Action pathway for lysine. But then you see the slinking arrow going back to spartate. It says feedback. So those of you who are in biochem, that should look vaguely familiar. Once lysine starts to accumulate, it feeds back to the first enzyme in the pathway and tells that enzyme, yeah, we got enough, stop. If you're trying to make a lot of lysine to sell, you don't want that happening, because you want lots of lysine. But if you do that without figuring this out, you'll reach a point that you won't get any more lysine because of feedback inhibition. So that's not good. So you need to find microbes that either can be tweaked or mutated. Excuse me. So that they don't do feedback inhibition. So this is something. When I say you got to get to know your microbes, but I mean these types of things. if you don't understand that this is going on. So, let's see, wait a minute. Do I have, well, maybe I deleted that due to time constraints. Let me just point this one out. The very first one, monosodium glutamate. MSG. That's made by microbes, and then they add it into food as a flavor enhancer or a meat tenderizer. But a lot of people are really sensitive to MSG. It's not the popular thing out there for you. So just be aware. But microbes are used for it. It's added to a lot of different foods. Okay. Exoenzymes. So remember now, when microbes are running their pathways to get their carbon, energy, electron sources, all of that, they're bringing in things from the outside. So basically what they do is they excrete out enzymes, break down those larger polymers, like a complex carbohydrate, break down starch, get down to glucose, then bring it in. So most of these enzymes we're thinking about with bacteria and fungi, they excrete them out. So they're called exoenzymes, technically. And the idea is that, they don't have, because of their small size, they have to break things down outside themselves in order to get the nutrients in small enough packets that they can easily transport them across the cell membrane. So amylases certainly are the starch. Exoenzymes are used to break down cellulose, which is, again, cellulose is just glucose molecules, but it's done in such a three-dimensional architecture that's it's very much a polymer. And so exoenzymes are used for that too. So here are some other exoenzymes that are commonly used in industrial practices: proteases, lipases, amylases. So you can actually take, I'm forgetting the name of the supplement, but there's a pill in pill form. There's a supplement you can take if you have, like, it's marketed something like, if you have an upset stomach, you can take these pills. And when you look at the label or ingredients, it's just a bunch of enzymes that microbes are using. And so I think the thought is that you swallow those, and those enzymes, if they make it into your gut, they help break down some of the food that's just making you feel not so good. But they also use these enzymes in laundry detergent, which I think that was a huge leap forward in laundry detergent when they started to do this, especially when they figured out, when you look at laundry detergent, it's got a high pH, it's above pH 7. So they had to go and find bacteria that live in alkaline conditions. And then they were able to extract the enzymes from those bacteria, put it into the laundry detergent, and the enzymes stayed good. They didn't break down. And so again, you have to know your microbe, you have to know your product. This is a really good example of, you have to go hunt for the right type of bacteria to be able to have enzymes that are adapted to alkaline conditions. And so Bacillus species, they can find several different types of Bacillus that are happy to live in an alkaline condition, and they produce enzymes that are quite good at staying active in alkaline conditions. So these, I don't think the detergents typically label that on the label. It's detergents, for some reason, they must have, I don't know, some kind of, they never show ingredients. Have you noticed that? You look on a detergent label and there's like no ingredients, other than they might say phosphate free, but they kind of keep everything hush hush. I'm sure you've heard of high fructose corn syrup, or I hope you have. And I say yuck to that because it's not good for your health. And unfortunately, that's something that is used through an enzyme action. So amylase and glucose isomerase that they get from bacteria were used in this process to create high fructose corn syrup. But again, not something you want to do and ingest if you can help it. And then this picture that you're looking at is a hot spring, I think at Yellowstone. And so this is the idea that when you go into really hostile environments like this, really hot or maybe super acidic, you get, they call them extremes, extremozymes. And so here, this is very hot. This is a hypothermophilic bacteria living in these types of hot springs. And that's where we get TAC polymerase from that you used in BI-218. So TAC, when you say TAC, that stands for the bacteria that they took it from. Thermus is the genus name, and Aquaticus, so TAC, T, and then the AQ, Aquaticus. So that's the actual bacteria that was isolated. And I have in the notes section that Kary Mullis is the story I heard is Kary Mullis had to go on vacation, and he drove up the coast of California, and he was trying to figure out a way to work with DNA, make copies of it, and come up with a way to do that. And he figured this out and put it all together. So he won the Nobel Prize for his efforts on this. So if you go to hot springs, there is a ton of life there. A lot of bacteria, some fungi hanging out there, and they're primarily responsible for the beautiful colors that you see. This is aerial footage of a hot spring. And so a lot of cyanobacteria are producing some more vibrant colors along the perimeter there. But there's other bacteria too, and some fungi. So they are there. And so you think about some of these hostile environments, and you think about some of the products that are made or processed that require, high temperature or, acidic situations or alkaline. So you have to go and hunt for these different regions and find your microbes that can handle that. Super salty, halophile. If you're a halophile, you can handle salty conditions. Yeah, acidophile, you can handle acid conditions. So they will add in microbes for poultry, food supplements for poultry. And so the enzymes, I should say, are added in as a supplement to improve their health. Psychrophile, it means you like cold conditions. And so sometimes you need either microbes or the products of microbes that can handle cold conditions. So I think the next slide gets a little bit at that. Yeah. So here, cold adapted enzymes, what can we use those for? And you can see a variety of different applications. Cheese ripening, starch hydrolysis, meat tenderizing. So again, lots of these are enzymes. Some of these same enzymes can be used at more like normal temperatures, but they've also found microbes that have cold adapted, proteases, for example. So it just depends on how you need to work with these enzymes and where you're going to hunt for these different microbes. Sometimes more room temperature is just fine, but sometimes you have to go into like the stone washing things that requires cold conditions. So we need to work with cold adapted microbes. Acetic acid. I'm sure you've seen that. Reaction pathway is in the image here. So when you want to make acetic acid, you start with ethanol. And the microbe will break down ethanol to acetaldehyde. And they use alcohol dehydrogenase for that. And then they use aldehyde dehydrogenase to take the acetaldehyde to acetic acid. So this is where if you ingest ethanol, and it's not so much the ethanol per se that is doing damage to you, it's more the acetaldehyde. That product is more, the product is more fiction. And so that Z-biotic product that I talked to you about where they have modified bacillus species to be able to break down the acetic acid in your gut. Because if that doesn't happen well enough, then the acetaldehyde builds up in your body, and that's more toxic than the ethanol. So it's not really the ethanol per se, it's the breakdown of ethanol that's causing problems. So acetic acid bacteria are very good at running that reaction. Now, if you look at vinegar, if you go and buy vinegar off the shelf, what that is the acetic acid, and then there's usually other microbial products in there too that they don't try to get rid of. So as they're running this in their production plant, they're using microbes to create the acetic acid, but of course microbes are going to excrete out waste products. And so the vinegar, that's when they start calling it vinegar. It's acetic acid plus other stuff. If it's just labeled pure, acetic acid, then it's just pure acetic acid. So vinegar is interesting. As far as I know, they don't make vinegar any other way. They use microbes to do that. And as you know, vinegar can be a very useful product for cleaning. Very useful in your kitchen, for example. The other idea is that ascorbate or ascorbic acid is also made by acetic acid bacteria. And that's actually vitamin C. When you look at the chemical structure of ascorbate, it's vitamin C, which we all need. Another product that you might see quite often on different ingredient lists is citrate or citric acid, same thing. And again, so that we're showing arrows going both ways because it goes back and forth like that. It's a fungus that they're using. Aspergillus niger is a fungus. And again, this is a case where you got to know your microbe because you can see the iron here. Third form of iron. So citrate is produced by this fungus when the fungus is sensing, uh-oh, I'm running out of iron around me. There's not much iron. I'm going to make a bunch of citrate, treat it out, so I can latch on this iron and bring that in. And so it's a chelating agent for iron. And so if you want them to make a bunch of citrate, you have to starve them of iron, essentially. And if you can do that, then they'll keep making the citrate. So again, once you get to know your microbe, you can optimize conditions so that it'll produce high amounts of citrate. And then, of course, fermentation. There's all kinds of products made through fermentation. That's how we've learned it. This is the scan of a book. Sander Katz is like the guru of fermentation in the modern world. This book is very thick. It's got anything you can think about in terms of fermented food product or drink product. He's got it all. And it's very easy to follow and read. People refer to it as the Bible of fermentation. So he's well versed, and this is a really well researched book. So their fermentation can be used for a lot of different things. Of course, we think about alcohol, which is on the list here, but there are other ideas that we use yeast for than just alcohol production. And again, it's usually through creating a situation where you make them ferment in one way or another. So certainly yeast used for baking bread, but certain vitamins as well, enzymes. So I wanted to put this in here too because this is gaining, like I said, it's gaining a lot of funding and research interest. There's a lot of companies that entrepreneurial companies that are popping up now trying to get into this area of research and development selling course components. So when you look at synthetic biology, it's tinkering with the microbe to kind of an extreme level. And so it's the synthesis of complex, usually biologically based systems, which display functions that don't really exist in nature. And so synthetic biology, they're trying to show that it's very interdisciplinary. You see all the different fields that you need people weighing in their expertise on this. And then on the right, you're seeing the application of what can we do with synthetic biology. You see that there's a lot of ways to spin this off into different applied fields. This is often how they talk about it, because you probably, the older term is biotechnology, but that's not quite the same thing as synthetic biology. So biotechnology is a little bit of just improving it a little bit. Something that in this case, the microbe does, and you just kind of optimize it or tinker with it just a little. That would be biotechnology. And that's been around for decades. But then with the advent of better technology, lower prices on things, so it was easier for more people to do, then it got into this idea that we could really start playing around with the microbes and do some really more impactful tinkering, I guess, where there's a lot more, for example, gene manipulation, gene additions, gene deletions, and looking at that, then you're wrapping into the synthetic biology idea. So that's the SynBio Beta is the company or the entity, the society that has that conference every year in California that I told you about. But they're really looking at exciting applications. So they're looking at the war on cancer, trying to figure out, can we use synthetic biology in that regard? I was very happy to see that they're implementing vertical growth systems as part of some of their efforts. But then they're actually looking at, things more, I guess, mundane things like cosmetics and fashion. So it runs a full gamut of how they're using synthetic biology in terms of what kinds of questions are they trying to ask and what kinds of things are they trying to solve. And so this is the idea that they're looking at, well, what happens if you grow microbes together? That's how they exist in nature. So maybe we can get some interesting products if we could grow them together. And so now this is pretty new. This is a pretty new concept, but it's exciting. They're coming up with this idea of, realizing that microbes work cooperatively to accomplish things. And if whatever they're accomplishing is what we want them to do, that's a good thing. And so this term SourceShot is showing up a lot in published papers looking at, where they'll talk about synergy, microbial synergy, and looking at that as well. Oh, I guess we got to stop. So there will be a few more things to say on synthetic biology, and then we'll get into exam review tomorrow as well, or Wednesday.